Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Cell Culture Basics: Initiate, Expand, Authenticate, and Cryopreserve Your Cells with Confidence, presented by Mr. Steve Budd. Mr. Budd is a product specialist at ATCC. In this presentation, Mr. Budd will tap into ATCC's vast cell culture experience and share the best practices for culturing cells that ensure optimal results and performance. He will cover all aspects of successful cell culture, including culture initiation, expansion, authentication, and cryopreservation. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Steve Budd.
Founded in 1925, ATCC is a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and service center in Gaithersburg, Maryland. We're the world's largest, most diverse biological materials and information resource for cells and microbes, the gold standard, which leads us into the topic of today's webinar on cell culture fundamentals. In today's presentation, we're going to start with cell handling tips, focusing on cryopreservation, aseptic technique and contamination, cell authentication, and issues with mycoplasma. We'll then discuss reasons for choosing certain cell types from primary cells to cell lines, and finish with some points on transfection and cell viability. So we're first going to start with some cell and media handling techniques. So to start with thawing, when ready to thaw a vial of cells, the most important factor to note is that unlike freezing cells, thawing cells should be formed as quickly as possible to remove the cryoprotectant from the cells. For best results, place the vial in a 37°C bath for approximately 2 minutes, gently agitating the vial to break up the ice. After removal from the bath, make sure to spray the vial with ethanol for decontamination. Transfer the contents to a sterile centrifuge tube with about 9 mL of complete media, which includes a cell culture medium and about 10% of FBS. When adding the growth media, pipette the liquid into the tube slowly in a dropwise fashion to avoid osmotic shock before adding the final full amount. When done, centrifuge the tube for 10 minutes at 125 Gs, discarding the supernatant and resuspend in 1 to 2 mL of growth media. Place the cells in the desired cell culture vessel with additional media. The main take-home here is to thaw cells as quickly as possible when placed in the water bath. Also note that for most primary cells, it is usually advisable not to centrifuge the cells, as this could be detrimental to the cell health. Simply plate the cells after adding the additional growth media. So after thawing cells, they should be plated in an appropriate vessel, which could be cell culture dishes or flasks. This is all depending on your specific needs. For most cell types, the general accepted practice is to allow them to settle for about 24 hours after seeding. At this point, the cells should be checked for confluency, which is the percent coverage of cells over the surface of the vessel. If expanding cells, the ideal time to cell culture is at approximately 80% confluency. The time it takes to reach this level can vary upon cell types. Primary cells can take several days or even up to a couple of weeks. The diagram gives a good visual of the different growth phases of cells in culture. When first plated, cells enter a lag phase where they grow slowly while trying to recover from the stress of subculturing. Cells then enter the exponential phase of growth. Here cells will grow exponentially as long as growth surface is available. When this surface is completely occupied, cells then enter the stationary phase of no growth. If they persist in this phase, they will then enter the decline phase, not shown in this graph, but to the right of the stationary phase. At this phase, they will lose viability and essentially die. To ensure phenotypic stability, cell lines need to be maintained in subcultures in the exponential phase. This will need to occur before a cell monolayer becomes 100% confluent or reaches its maximum recommended cell density. As stated before, that ideal confluency is about 80%. So when you've reached 80% confluency, cells can be passaged with trypsin EDTA. Trypsin disrupts the proteins involved in attaching cells to the culture vessel. The figure on the right shows a progression of cells being trypsinized. At the starting point, we see cells attached as a monolayer. After trypsin is added to the cells, they become flattened and more round. When completely detached, they can be seen floating around in clusters. This should take about 3 to 5 minutes, but make sure to note that the dish should be gently agitated to help detach the cells, otherwise it could take longer. Long-term exposure of trypsin to cells can be detrimental, so that time in trypsin needs to be minimized. Also note that if using primary cells, a special lower concentration of formula of about 0.05% trypsin should be used. The lower concentration of trypsin will definitely require the physical agitation of the cell culture dish to ensure detachment. Also, when trypsinization is complete, the reaction needs to be quenched, ideally with a soybean neutralizing solution. Again, this will help protect the cells from any damage of long-term exposure to trypsin. In the next several slides, we want to discuss techniques on cryopreservation. I want to first go over the basic theory behind cryopreservation and how it works. So the freezing process is a complex phenomenon that's been studied for decades. Cellular metabolism stops when all water is turned to ice. This, however, is largely detrimental to the cell's health. Two main reasons for this: ice formation and an increase in solute concentration within the cell. This is caused by either freezing too quickly or too slowly. The solution to this problem is maintaining the ideal freezing rate while using a cryoprotectant, the most commonly used being DMSO. The combined effect will minimize ice crystallization and reduce the solute concentration effects. An important take-home here, as the diagram notes, for most cells, the optimal rate of freezing is a drop of 1 to 2 degrees Celsius per minute. So freezing cells is typically a two-stage process in which cells are incubated for some time at minus 70 degrees Celsius before being stored at minus 140 degrees C. As previously discussed, cells must be cooled at a rate of about minus 1 degree Celsius per minute. This can be achieved with the use of a controlled rate freezer or controlled rate chambers. The length of time needed in the minus 70 will depend on the type of chamber used. So after spending the appropriate time at minus 70 degrees C, the vials must be transferred to minus 140 C for long-term storage. It's recommended to store cells in liquid nitrogen tanks for this purpose. So as I just stated, long-term storage of mammalian cells require that cells be exposed to temperatures below minus 140 degrees Celsius. At these low temperatures, metabolic activity ceases, allowing for the preservation of cells for an indefinite period of time. For storing biological material at these low temperatures, it is highly advisable to use liquid nitrogen freezers. So there are two basic types of liquid nitrogen storage practices. One is immersing the vials directly in the liquid. The other involves holding the vials in the vapor phase above the liquid. Vapor phase systems create a vertical temperature gradient within the container. At the very bottom, where the liquid nitrogen resides, the temperature will maintain about minus 196 degrees Celsius. The temperature in the vapor nitrogen decreases as it reaches the top portion of the container. Enough liquid nitrogen should be used to guarantee that the warmest part of the container at the top is always at a temperature at or under minus 140 degrees Celsius. It is highly recommended that vials be stored in the vapor phase rather than the liquid. Since metabolic activity is arrested at minus 140 degrees C, the additional cold temperatures at the liquid phase offers no real advantage. However, immersing the material in the liquid phase runs the risk that some of the liquid nitrogen could penetrate the vials and cause them to crack or even burst. As we leave the discussion on cryopreservation, I want to discuss cell characterization as it relates to our upcoming topic of general cell health, contamination, and authentication. It is necessary to characterize cells to determine how healthy and viable they are, as well as making sure that they are the cells you think they are. Simple cell viability counts using trypan blue with the hemocytometer are an easy way to determine the percentage of live cells over dead cells. With this method, live cells appear to have bright centers while dead cells appear dark blue as the dye penetrates the compromised membrane. Often when researchers receive cells from collaborators or other unreliable sources, they may find that the cells are not exactly what they thought they were. So morphology is a quick way to distinguish broad cell types from one another. In the example on the right, you have two cell types, fibroblasts and HUVECs. They have very distinct morphology. Fibroblasts tend to be elongated, whereas HUVECs tend to have a rounded cobblestone appearance. Also, since different cell types grow at different rates, always make sure your cells are growing the way you would expect. If cells are contaminated with another fast-growing cell, the growth rate and doubling time may be greatly increased. We now come to the topic of contamination. So the main types of contamination we'll be concerned about are microbial, which include bacterial, mycoplasma fungi, and virus. There's also cellular contamination, meaning one cell type or cell line has invaded another. We'll discuss this in a little more detail shortly. So bacterial and fungal contamination are the most common problems seen in labs. They often appear quickly, but fortunately can be detected very easily. Turbid and acidic media usually indicate the presence of bacteria. Cell stress from microbial contamination may also lead to morphological changes in the cell type. Fungal contamination is seen by the appearance of obvious filamentous structures in the media. So at least one good thing about this microbial contamination is that it's very easy to detect quickly. Mycoplasma and cellular cross-contamination, discussed in just a minute, is not as easy to detect. So where does contamination come from? For starters, anything that has been contaminated in the past can contaminate cells going forward. Media and reagents that are not handled correctly can easily be a vehicle to introduce bacterial and fungus into cell cultures. Using items such as petri dishes or pipette tips that are not properly sterilized can certainly introduce contaminants. Personnel can also introduce particulates and aerosols on personal clothing or even on dry skin. Improperly maintained equipment can prevent protection from the entry of contaminants. Overpacking autoclaves or ovens can even cause problems with uneven heating, creating pockets of non-sterile supplies. The flow rate of hoods, if not properly adjusted, can allow particulates to enter. So what can you do to make sure cells and cultures do not get contaminated? So I'm going to point out a few tips that should prevent contamination problems. First, in general, you want to make it hard for contaminants to enter your cell culture vessels. Small dishes, such as 35 to 60 millimeter dishes, can be placed in larger ones when in the incubator. Placing plates and dishes in larger ones are also ideal for transport. This adds an additional physical barrier that prevents particulates from entering. When using flasks, it is recommended to use vented caps that can be sealed tightly but still allow gas exchange. I always use disposable aspirators and pipette tips that are pre-sterilized. Make sure that your hood has adequate laminar flow and never overcrowd or leave materials such as boxes or bottles in the hood indefinitely. This can impede proper flow and block attempts at cleaning and wiping down the hood surface. When bringing media and reagents into the hood, always spray the items down with alcohol to remove any contaminants. It is also advisable to bring in small amounts of reagents into the hood at any given time. If possible, you may want to aliquot small amounts in these containers to reduce the number of times you bring a possible contaminant into the hood. For instance, if a vial is contaminated, at the very least, it won't continue to enter the workspace. Always remember to wear clean lab coats that cover most of your arms to prevent dust and shedding skin to contaminate your cultures. We advise not using antibiotics in culture. There are a few reasons in addition to the potential of toxicity. If you use media with no antibiotics and a contaminant enters your cell culture, chances are it will be visible within a couple of days. You can then discard the infected dishes. However, if there are antibiotics present, it may slow the growth of the contaminant, allowing further passages and freezing down your cell lines. In short, you may be saving large amounts of contaminated cells that can cause problems down the road. Resistant mycoplasma may also get introduced along with other bacteria. The antibiotics may get rid of the larger bacteria, but mycoplasma may persist undetected. So in short, antibiotics can also sort of cause this sort of sense of false security. When is it acceptable to use antibiotics? So a few exceptions make it necessary. For instance, if you're isolating primary cells from original tissue, it may be a good idea to use the antibiotics for the first couple of weeks. So Mycoplasma contamination is a separate contamination issue that is extremely difficult to detect. So contamination from mycoplasma can cause a host of problems that includes altering the cell's function, growth, morphology, causing chromosomal aberrations, affect nucleic acid synthesis, and changes in membrane antigens and gene expression. The main source of mycoplasma contamination is receiving infected cultures from other research laboratories. Reagents purchased commercially can also be a source of contamination. Mycoplasma contamination can be detected through a number of available kits in the market. The Universal Mycoplasma Detection Kit from ATCC is a PCR-based kit that can detect 60 of the most commonly identified Mycoplasma species. So to put it in perspective, over 90% of contamination is caused in just a few species. The Mycoplasma Detection Kit comes with specific primers that are unique to Mycoplasma with Mycoplasma DNA as a control. To test for Mycoplasma, simply collect and lyse cells from your culture, perform a touchdown PCR, and run the product on a gel. If the cell culture is contaminated, a band will appear at the same location as a positive control. Cross-contamination of one cell type with another is also a problem. If the contamination is a particular fast-growing cell, the original cell line can be gradually replaced with the invading cells. As a result, an entire cell line in one's depository can be completely overwritten with a new cell. Furthermore, morphology is often not reliable enough to distinguish between the original and invading hosts, particularly if they're the same cell type, like one endothelial cell invading another endothelial cell. So there are a few common causes for this kind of contamination. Researchers can contaminate their own cell lines by working with multiple cell lines at a time. This often occurs when using the same pipette tips or aspirators with vessels containing different cell types. We recommend only working with one cell line at a time under the hood if at all possible. More commonly, researchers receive cell lines from other labs or companies that were already cross-contaminated, so the importance of this cannot be understated. It has been estimated that 20% of scientific publications use misidentified cell cultures. This has also resulted in about 50% of preclinical research being non-reproducible. As a service, ATCC offers cell line authentication utilizing short tandem repeat profiling. STR profiling helps to detect misidentified, cross-contaminated, or genetically drifted cells, which invalidate research results. To take advantage of this service, simply receive a sample packet through the mail, spot cells onto a paper insert, and mail back. You will receive a report that includes the STR profile of your cell line, a comparison of your cells against an STR profile database, electropherogram supporting the allele cause at each loci, and a complete interpretation of the results. I also like to note that when you get back into the lab, now is a good time to start with freshly authenticated cells, and we do now offer human and mouse authenticated cells. So here I'm going to change focus and go into how you can choose the best model. When we're talking about which best cell model and why you would want to choose this. But before you go into that, I want to briefly discuss media before you go into the models. So in general, continuous cell lines use a classical cell culture medium, such as DMM, RPMI, 1640, etc. usually with 10% FBS. More specialized cell types like primary cells, H-chert cell lines, stem cells, tend to require more specialized media. It is generally recommended to maintain cells in the same medium, but if transferring to a new medium, it should be done gradually with the 50-50 mix of old and new, eventually reaching the new medium entirely. ATCC does not recommend using heat-inactivated FBS under normal circumstances because it can denature growth factors necessary for the health of the cell. There are several types of cell models to choose from when designing your experiment, each with its own set of pros and cons. So let's start with the far right, the continuous cell lines. Continuous cell lines are often derived from a tumor. They're generally very easy to culture and have a low cost of culturing. They do not require a complex growth medium, usually just one of the classical cell culture formulations like DMM or RPMI supplemented with FBS. Like a tumor, they have great proliferative capacity. They all originate from a single clone, so there's minimal inter-experimental reproducibility. However, they are by nature abnormal cells with high instability and thus provide low in vivo predictability and limited biological relevance. The next model, cell type that I would like to discuss is H-chert immortalized cells. These cells are genetically modified primary cells such that the cell no longer undergoes senescence. So what it's created is a cell with continuous cell-like growth characteristics but primary cell physiology. This means high proliferative capacity, and since every H-chert cell type originates from a single transfected clone, there's no inter-experimental variability. Every lot will be virtually identical. Since the starting material is a normal primary cell, there is greater genetic stability with near normal primary physiology, so therefore high predictability and high biological relevance. The third cell type is an induced pluripotent stem cell derived primary cell, or iPSC derived primary cell. This cell type is derived from a somatic cell line that is dedifferentiated to an embryonic state and then differentiated to a specific primary cell lineage using a specific cocktail of growth factors. iPSC-derived primary cells are usually generated from a single clone, so there's likely little worries regarding the introduction of biological variability. The main problem with stem cell-derived cells is that they are differentiated in the immature primary cell types, so not the best for predictability or biological relevance. Lastly, we'll discuss the cell type that is the best model for in vivo predictability and biological relevance, the primary cell. A primary cell is usually isolated from normal adult tissue, so no genetic instability. However, for large-scale screening, there is a potential problem, not enough cells from a given donor. The use of new donors will introduce biological variability in your experiments. So each model has its own strengths and weaknesses. However, as you move from basic research to more clinical-like applications, you need to choose a model that most positively matches up to your research needs. As a final note regarding choice of cell model, continuous cell lines are derived again from a tumor or mutated in some fashion, such that the cell has deviated from its original source. So in every experiment involving continuous cell lines, the corresponding primary cell should be run as a control. That way, if you observe a specific effect on your experiment, you can be sure the results are valid and not an artifact of the tumor or mutation. So the take home is use primary cells as controls when at all possible. Okay, we're going to switch gears again and talk a little bit about transfections and what transfection reagents you can use. So now I'm going to discuss the basic method of lipid-based transfection. Since transfection is a method for introducing exogenous nucleic acid sequences into cells, there are various ways to accomplish this. One of the most common is lipid-mediated transfection. Lipid-mediated transfection is the most accessible and usually the simplest method available. It's based upon the idea that opposites attract. The transfection reagent is a cationic lipid that forms micelles with a net positive charge. Nucleic acids have a net negative charge, allowing the formation of complexes of lipid and DNA. The nucleic acid is covered by the micelle, so the complex now has a net positive charge on the outside. The surface of a cell has a net negative charge, so the complex moves to the surface and binds to the cell membrane. The nucleic acid lipid complex can now move through the cell's lipid membrane, allowing the nucleic acid to enter the cell cytoplasm. The nucleic acid then enters the nucleus, and the gene product can be expressed. ATC supplies two lipid reagents for transfection, GeneX Plus and Transfex. Transfex is our all-purpose reagent working well with continuous cell lines in primary cells. As suspension cells are a bit more challenging for transfection, we often find that GeneX Plus offers higher transfection efficiency for these suspension cells. So it's very easy to actually set up a lipid transfection process. And here I'm going to go through a simple workflow to demonstrate this. You always want to transfect cells when they're in the exponential growth phase. So you seed your cells to be ready the day before you plan to do your transfection experiments. On transfection day, which is day 0 on the slide, you combine your DNA with your lipid reagent and allow the complexes to form. Then you can add the complexes to your cells that you seeded on day -1. It's at the very top of the chart. You should then be able to detect protein expression at about 24 to 72 hours post-transfection. So now we're going to sort of talk about transfection best practices and how to optimize culture conditions. And these are 4 things to take into consideration. So even though it is a fairly straightforward protocol, there are several aspects of the process that you need to be optimized to ensure the highest potential, the possible transfection efficiency. To achieve a good transfection, the health of your cells is of utmost importance. The cells should preferably be at a low passage number. If they're seeded too high or too low of a density, the metabolic rate could be negatively affected, and cells could find the complex as toxic or may not readily express the gene product of interest. Contamination can lower the observed transfection efficiency, especially if it's mycoplasma. If you ever observe a drop in efficiency with the transfection protocol that has been previously optimized, Mycoplasma contamination may be a likely reason. Media used during the transfection process is also a consideration. Certain lipids require no or low serum during the transfection process, so always follow the lipid manufacturer's guidelines. So a little bit more on transfection best practices. So for nucleic acids, which are DNA or RNA, one should use high purity and endotoxin-free material to achieve the highest transfection efficiency possible. Endotoxin appears to lower transfection efficiency. For DNA, one should verify promoter strength for the given cell being transfected. Certain promoters are known to work better with certain cell types. Plasma size is also a concern. The larger the plasmid, the lower the expected transfection efficiency. You should also verify that you are using a complete sequence in your plasmid. DNA plasmids that have been stored for some time can become degraded or nicked. So when designing your transfection experiment, you need to determine what assay methods you will employ at the timing for the data collection. As your protein of interest is expressed, you can use real-time PCR to detect RNA, probably 18 to 48 hours post-transfection. If this is your first time working with proteins, I would suggest running a time course analysis at a few different time points. If you plan to detect at the protein level, you need to give the cells enough time to make your protein and then perform any analysis before the protein is turned over. If you're not sure of your protein's half-life in the cell, again, a time course analysis is warranted. So of course, depending on the nature of your protein, there may be additional functional assays you can run. However, I would always recommend running analysis at the genomic and protein level, at least until optimal transfection parameters have been established. So regarding transfection reagents, there are three main areas of concern. First, which lipid should I use? That would depend on the cell type you're using and the cell characteristics. As I just mentioned, certain lipids are generally geared towards cell suspension, like ATCC's Gen X+. Other considerations involve what are you trying to deliver to the cells. Certain lipids are for DNA, others may be designed to work with RNA or small proteins. Next, you often have to optimize the ratio of lipid to DNA for the cell type and substrate that you're working with. Most manufacturers will provide guidelines, but some optimization should be expected. Lastly, one should optimize the incubation time for the lipid in contact with cells. If you're using a low or no serum transfection medium, you do not want to leave the cells in the medium for too long, and some complexes can become toxic to cells. So again, most suppliers will provide guidelines, so optimization may be required for your specific conditions. And so this is the very last slide on transfection. I'm just going to summarize best practices on the transfection section of this talk. So to achieve optimal transfection efficiency, use healthy cells in the exponential growth, free of contamination, DNA that is of high purity and endotoxin free, and the recommended lipid for each cell type and substrate should be determined. Every setup may require some optimization, including but not limited to seating density of cells and the ratio of lipid to substrate. And so now we're going to move to the last major topic of this discussion, and this is going to be viability assays. So to cover growth and viability, we want to discuss ways of actually measuring the number of cells in a cell culture vessel and determine the proliferation or growth rate. This is ideal for assays in which you want to compare normal growing cells against an experimental factor that would positively or negatively affect the growth. So these two assays that we offer, MTT and the XCT cell proliferation kits, utilize tetrazolium salts to deliver a quantitative analysis of cell numbers. So this slide is a diagram that shows how this works inside of a cell. So you can imagine the blue spheres are cells, and the gray inner sphere is going to be the nucleus. So with the MTT assay, the tetrazolium salt is introduced into the cell. Naturally occurring oxidation reactions within the cell, which is indicative of normally growing cells, will reduce the tetrazolium into formazine. So the cells are then lysed with detergents, releasing the formazine, giving off a purple color. The more purple the media, the more metabolically active and growing the cells will be. So this assay is good for measuring things like cytotoxicity and apoptosis. And the XCT reaction on the right is very similar, but the reaction takes place at the surface, and it gives off an orange color as an indicator. From a more practical point point of comparison, so both assays are designed for 96-well plates, and here we have an overview of what these assays are going to look like. So for both, they're plated with, in this example, with a cytotoxic test compound. With the MTT reagent at the top, there's an incubation period to allow the reagent to enter the cell. The detergent is added, allowing for the compound to be released in the media. At this point, it can take a few more hours for the reaction to complete. With the XCT reaction, the XCT reagent and activation agent are added at the same time, so the reaction starts immediately. So one just practical benefit of the XCT kit over the MTT is there's one less step in the protocol, which will knock off a couple of hours from the whole process. And so this graph shows an example of absorbance versus cell number when comparing the MTT kit and the XCT kit. The XCT is in the blue at the top, the MTT in the red at the bottom. So in this case, eventually the absorbance will saturate and no longer indicate the number of cells as the number of cells increase in your plate. So the reading is taken on the growth curve. So this shows that with the XCT kit, it's more sensitive than the MTT, and that they can read cells between 5,000 to 30,000 cells. So with the MTT, the reading is between 5,000 and 15,000 cells. So the XCT assay, because of its fewer steps in the protocol and its increased sensitivity, is generally considered to be a more sophisticated assay. And so now we're reaching the end, and this is just one last summary on everything that we've talked about. So as a summary of what we've discussed, remember to use best practices to eliminate contamination while ensuring optimal growth and storage. In your cell culture workflow, choose the best model for your needs. Cell lines for standardization and primary cells as optimal controls that maintain biological relevance. H-turn immortalized cells can be the best of both worlds with continuous growth and primary cell characteristics. And so the key to good transfection practices rely on the use of healthy cells, purified substrates, and optimization of the lipid to substrate ratio. Lastly, you can monitor cell health and viability with the MTT and HTT viability assays. And that concludes this webinar on basic cell culture handling. And I will turn it over to Dr. Shapiro for further announcements.
Well, thank you, Steve. In just a few moments, we will begin our Q&A session. Please remember to use the chat function available through the webinar program to submit your questions. And just as a reminder, the recorded webinar presentation, as well as the slides, will be available on demand on the ATCC website at www.atcc.org/webinars. And it looks like Mike Utaro, one of our technical service representatives, has joined in. So welcome, Mike.
Hi, Brian. Great to be here today.
Okay, well, we've had a ton of questions come in today. Let's go ahead and start with our first one. And I'm going to send this to Mike. Can we use contaminated culture after treating that culture with antibiotics?
Okay, a good question. So generally speaking, it's never really recommended to use cells after they've been contaminated or after you've noticed a contamination in the culture. In addition to being difficult to get rid of, cells could have been physiologically compromised from exposure to the contaminant. And so in general, we definitely recommend getting rid of the culture, definitely trying again, if this can be very difficult to try and save.
Gotcha. Okay, so this is next question it's a great question. It certainly came up many times for me when I was a grad student. What are the main reasons cells may unexpectedly detach from the culture vessel?
Okay, so yeah, there are definitely a lot of reasons why cells could detach unexpectedly. More often than not, this is going to be due to suboptimal pH in the media and incorrect buffering. This could be from an imbalance of the carbon dioxide percentage in the incubator and the amount of sodium bicarbonate present in the basal media, as well as any other differences in, say, like if you're using a different formulation from ATCC, it could have different concentrations of HEPs or other buffering agents. So that's definitely one of the things that could be playing a role. So most ATCC formulated basal media will contain 1.5 grams per liter of sodium bicarbonate, and that's going to be optimized for 5% carbon dioxide. When using alternate media formulations like from Gibco or from Sigma, most of those are going to be formulated with 3.7 grams per liter of sodium bicarbonate. That level of sodium bicarbonate is going to be better suited for 10% CO2. And so that increased amount of CO2 that's necessary when using that 3.7 gram per liter sodium bicarbonate is going to be necessary to make sure the cells attach well and don't detach during culture. Confluency density of the cells in the flask could also be playing a role. If the cells are too dense, they could start lifting off of the flask because of lack of substrate to attach to. And then finally, when you're subculturing, the time of exposure of the dissociation enzyme, whether it be trypsin or any other enzyme, incomplete inactivation of these dissociating enzymes And cell clumping can also have an effect on cell attachment later on.
Alrighty, good, good. Now this next question, it's a specific product question about the Mycoplasma Detection Kit. So I'm going to send that one over to Steve. Can you tell us how many samples can be run with the Mycoplasma Detection Kit?
Sure. If you follow the protocol the way we suggest, if you follow our protocol, you can run about 12 samples. It's about 40 reactions in the kit, but we suggest you run each sample twice as duplicates. The third is a positive control with the sample. You want to run the positive control of the sample to make sure there's nothing in the media that could interfere with the PCR reaction. So there's also the positive and negative samples in there. So basically, with all the different controls and duplicates, you can run about 12 samples per kit, 12 individual cell types.
Okay, yeah, that makes sense. You want to be sure that you have your positive control and duplicates with every test that you do, right? Okay.
Correct.
So this next question that I'm going to shoot over to Steve as well. Can you keep the freezing container, the cool cell, for long periods, like several days or weeks? And I think what they mean specifically is, can they keep it in the minus 80-degree freezer for longer than overnight, which is what's recommended?
For the cool cell in the minus 80, we recommend no more than overnight, no more than 24 hours. It can be as low as 4 hours. So you have a pretty good window there that if you want to freeze your cells down, say early in the morning, you can only put them in there. You can put them in there for 4 hours or so, and then later in the afternoon put them in the liquid nitrogen. Or if you are running them later you want to freeze them down later in the day, you can certainly put it in the minus 80 overnight. But we recommend no longer than 24 hours in the minus 80.
Okay, yeah, that makes sense, or else you're going to risk cell damage, right?
Correct.
Okay. Now this next question I'm going to send Mike's way, although Steve, you did cover this in your talk. So Mike, does liquid nitrogen tank with stored cells, okay? Does it have to be filled almost to the top with liquid nitrogen? Or to maybe ask a different way, do the cell culture racks within the nitrogen storage unit need to be above or below the level of liquid nitrogen?
Okay, so filled to the top is going to be far too much liquid nitrogen. That is definitely going to be overkill, for lack of a better word. So when we say store in liquid nitrogen, we do mean store in the vapor phase of liquid nitrogen. It's generally not advised to submerge cell vials inside liquid nitrogen, mostly because any sort of cracks or openings in the cells, that liquid nitrogen would be able to get into the vial. And then when you take it out of the liquid nitrogen, that's just going to rapidly expand and could cause an explosion. By explosion, I mean the vial exploding. So in terms of storing cells, you should have enough liquid nitrogen in there to keep the tank cool, but you definitely want to store cells in the vapor phase of liquid nitrogen and not submerged inside liquid nitrogen.
Right. I can remember pulling, when those are overfilled, you pull them out and then liquid nitrogen pours all over the floor and that sort of thing when someone fills it up to the top. And it's also kind of a waste of liquid nitrogen too, right?
Oh no, absolutely, because you definitely do not need you don't need a completely full tank. That's definitely that's an excess that is absolutely not needed.
Now, and this is another question that we get a whole a lot, but it's kind of related. How long is a cell line stored in liquid nitrogen viable? So, like, say you buy a cell line from ATCC and you can't thaw it and grow it immediately, and you put it directly in liquid nitrogen storage. How long can it be stored without losing its viability?
Yeah,
So,
I can answer that.
go ahead.
Yeah.
Go ahead.
Oh, no, thanks. Yeah. Pretty much all cells stored in liquid nitrogen there's no expiration date. It's indefinite. At that point you're at a point where all molecular activity ceases. So there's no theoretical time limit to how long it can be stored there. It's indefinite. We've had things stored in liquid nitrogen for decades and recovered them.
All right. Good, good. Now, wow, we've got a whole lot of questions come in. Let's go ahead and jump to the next one. Can we use treated T25 or T75 flasks for suspension cells? I assume that means treated for cells to adhere. Can you use that for a suspension cell? And I'm going to shoot that to Mike.
Yeah, I mean, that shouldn't be a major issue. So with most suspension cells, you are going to see sometimes one or two, a few cells attaching. It's you shouldn't really see the cells. If the cells are designed to be in suspension, using a culture-treated flask is not going to cause them to adhere, at least, to the level that you'd see for other adherent lines. So it should be fine. If you really are concerned about cells adhering, there are flasks that are sold that are specifically designed for low attachment. So it's definitely worth looking into if you are concerned about that. But in general, it should not have any sort of issues when using a suspension cell line with cell culture treated flasks.
All right. I like it. Okay, so this next question, I guess I'm going to ask you to elaborate, Mike, a little bit on an answer that you gave about cells detaching. So could you elaborate on CO2 versus bicarbonate for use for buffering?
Okay, so CO2 and bicarbonate, they work together to create the buffer in the culture media. So CO2 generally is going to react with water in cell culture to create carbonic acid. By having bicarbonate there present, that kind of helps prevent that from kind of occurring rapidly, keeping the media pH of the media at that neutral range while working in a CO2 incubator.
Okay. Now this.
So basically, actually.
Oh, sorry.
Sorry, I wanted to touch on that. Just one more quick thing about that. So there are media that doesn't include sodium bicarbonate, things like L15. Those media can't be used in carbon dioxide incubators because they don't have the bicarbonate, and so incubation of that media in a CO2 incubator is going to cause that media to rapidly acidify, and it's going to kill off the cells.
Okay. Good. All right. Now this next question, it's actually a little bit more of a microbiology question than a cell biology question. So Mike, in some E. coli urea broth cultures from a pooled shRNA screen, I'm experiencing some non-pelleting in some of the 5 ml tubes. On examination with the naked eye, centrifuge tubes appear to contain a yeast or endocytic fungus of some kind, possibly Candida. This will not pellet like the bacteria. Have you ever heard of such contamination in Ampicillin LB E. coli cultures?
So I have not heard. So if ampicillin is being used, that should preclude a lot of other contaminants from even LB broth. As far as, the lack of pelleting, if the culture's not growing and you're not seeing what you're seeing seems to be, completely atypical from what you're used to, it may be worthwhile just to discard those tubes because it sounds like it may not be a contamination per se, but it doesn't sound like you're experiencing growth in those tubes. Uh, and with E. coli, you should hopefully have a additional somewhere. It may just be worthwhile to create new tubes, especially if you're not seeing, pelleting to extract shRNA from.
Okay. All right. Now, sort of a follow-up contamination question. Have you ever dealt with black dots that seem to be non-living or inorganic particles? And, how would you handle that?
Okay, so black dots. So the first thing you're going to want to see is look at it under the microscope, and if the dots aren't moving purposefully, if they don't look like they're swimming in, purposeful directions, things like that, it could very well just be cell debris. That's definitely not an uncommon thing to see in cell culture. If you are concerned about the black dots, you can always take some of the culture media and add it to just a fresh flask with just media in it to see if the black dots grow, or if it is a contamination, it will grow very quickly in that media. But black dots present, it's not a major concern. It could just be cell debris, and if that's the case, slow centrifuge speeds can definitely help clear that up a little bit.
Okay. Trivia question. What about if your culture turns black? Is there any opportunities where your cell culture might turn black for some reason? Not opportunities, but instances.
I can only think of one instance off the top of my head, and that would be working with melanocytes from dark pigmented donors. Black is not a color that you typically see in cell culture, and if you're not working with melanocytes and your culture is, a black color, I would be concerned.
Right, and that's exactly what I was shooting for. Yeah, that would be, if you're working with melanocytes, they make melanin, which is a pigment, and that can definitely make your culture medium turn black.
Mm-hmm.
All right. So this next question, I guess either Steve or Mike can tackle. Do you consider the use of a face mask as a critical primary barrier to prevent Mycoplasma contamination? Presumably, this is if you're working with a biosafety cabinet.
Yeah, I'll just jump in and then let Michael give any more detail. It's a good precaution, but as long as the biological cabinet you're working with has the proper flow rate, that should be sufficient for really any contamination issue. But I'll let Michael say anything about that if you'd like.
No, you basically covered it. I mean, especially when you're working in a biosafety cabinet, the glass should be, below your breathing level anyways. So the risk of you breathing, in the biosafety cabinet should be low. And again, the flow rate, like Steve alluded to, that should hopefully preclude, any of your breath getting inside the biosafety cabinet if it's calibrated correctly.
Yeah, that's what I thought as well. Okay. Here, this next question is kind of interesting. Is it possible to identify a cell line using flow cytometry? And actually, I feel like I can kind of take this one and give you guys a rest. So, Mike, honestly, I would say no. you could probably identify what type of cell it is via the biomarkers it has, right? Whether, it's T7 or von Willebrand factor, or cytokeratin, what particular type of cytokeratin it's expressing. You could determine if it's, an endothelial cell, a fibroblastic cell, or an epithelial cell, but I don't think you could specifically say, that it is like that's an A549 cell right there,
No,
so
exactly.
So this next question, let's jump over to this one. This is about cryopreservation. Do you know whether the FBS percentage in the freezing media can affect cryopreservation?
So effect is a pretty broad term. I can't think of any negative effects that would come from using a high FBS cryopreservation media. There are cell lines that will use 90% FBS, 10% DMSO. I can't think of any, effects one way or another. Some cells are going to freeze better in 90% FBS. Some cells are going to freeze better in complete culture media. Yeah, it's hard to say like what kind of effect, if any, FBS would have on the percentage of FBS could have on the cryo media.
Yeah, and I'll just go on that.
Oh yeah, go ahead, Steve.
Oh yeah, sorry. Yeah, I was just saying typically most protocols say to use the complete growth media or growth media which has 10% FBS in it and the DMSO, which brings it down to about a final concentration of 10% FBS or less. Anywhere from 5 to 10% is what I think a lot of cell lines in primary cells use. So if you're using a good using growth media with that is very very optimal for a lot of situations.
Yeah, for at least all the cell lines ATCC provides, we have the protocol with the recipe to use, right? I would just basically use whatever that is, and that was determined empirically by whoever posited those cells with us. So, yeah.
Yep, yeah, correct.
Okay. Now let's see. Can we select any type of tumor cells for transfection? I guess I'll send that to you, Steve.
For transfection, or at least the ones we use, there are certain cell types that are ideal. If you mean as a transfection vector, there are certain types like our transfection reagents. We have certain protocols that have been optimized for certain cell lines. So we would advise using our transfection reagent, using those optimized protocols that are specific for those cell lines. If you want to add anything to that, Michael or Brian, yeah.
Yeah, I'd have to take a look on the website, but I know that we have 2 transfection reagents, right? And
correct.
Pretty sure that one of them seems to work well with fibroblasts. So I would have to take a look at the Adam. That's what Transfex and Genex Plus, right?
Correct. Yeah, absolutely.
Yeah, and each of them have protocols for the cells that we've tested with them that to see that if they work. So all right. Let's see. If ATCC doesn't recommend use of heat inactivation, then how does the cell get rid of complement system as a potential threat for survival? That's kind of an interesting question.
So most cell lines actually aren't going to be negatively affected by the complement that's seen in fetal bovine serum. It's mostly going to be a concern for, I would say, some of the immune cell lines, B lymphocytes, T cells, things like that. But for the most part, most cells aren't going to be negatively affected by the complement. And yeah, so I can't really say about how they will get rid of it per se, but I know that pretty much for most cell lines, it should not have any sort of adverse effect on the cells.
Yeah, I would agree with that. You know, if you're bacteria, yeah, you should be worried.
Yeah. And one thing to note, heat inactivation can, degrade some of the growth factors and components that cells need. The whole reason why you need the FPS in the first place. So it can have a negative impact on that, on the growth factors and reagents in the FPS.
Mm-hmm. So we definitely avoid using it unless it's specifically called for. But cell lines will kind of have that specifically labeled on their webpage if they need heat inactivated FPS or heat inactivated serum for that matter.
Yeah, exactly. All right. Okay, this next question's around the DMSO used for cryopreservation. Does it need to be sterile-free filtered?
Yeah, I mean, it should be sterile. You don't want to use dirty DMSO when you're freezing your cells. DMSO that ATCC sells is going to be sterile filtered. Most DMSO that you get from most commercial entities are going to be high quality, already pre-sterile, but yeah, you should be using sterile DMSO when you're freezing your cells.
Yep, I would definitely agree. And by the way, that ATCC part number is 4F for DMSO. All right. Here's an interesting question. There's a couple ways to look at it. But we have multiple vials of frozen down cells, which all came from the same batch of cells. So it sounds like someone's adhering to the seed stock principle here. Some vials grow up to their 30th passage with no problems, but we have random times where cells begin to round up and die within 4 passages of thawing. Any idea why this could be? How to test for this beforehand, maybe? And they said that the cell lines that they're using is U2OS.
So, the first thing that would immediately come to mind about vials from a same freeze down that aren't growing well are, is it being handled by the same researcher? Are the reagents the same? Is the media the same? Protocols the same? Things like that. That would be the first thing that I would immediately want to look into. Mycoplasma contamination is also one that I would want to bring up. I don't want to say it's likely, but it is something that could explain, one flask not doing well after a few passages versus other flasks, as you've noted, that, can get to passage 30 and above. Some vials, especially when you're doing a large freeze down, sometimes some vials just may not revive well. It's kind of hard to say because, it sounds like you're able to get viable cultures from the freeze down that you've done, and there are definitely a lot of factors that could be affecting recovery of the cells, different vials of the cells from the same freeze down.
Yeah, there's one other thing that kind of jumps out at me is, the theory of cancer heterogeneity, where not every single continuous cell that's within like one culture is going to be, the exact same as every other, right? And so you may, through aliquoting your cells, get some that have cells that just don't grow or perform nearly as well. But I'm gonna say it's probably like through some inconsistent practice, like Mike's talking about.
That would definitely be, start with the, I guess, Occam's razor. Start with the most likely. Start with protocols, reagents, and then kind of work backwards from there.
Yep. I like it. All righty. Let's see. So do reagents used in viability assays like MTT or XDT interfere with pH indicator in the media, such as phenol red?
To sort of reverse or sort of to answer along the lines of that question, if the pH of the cells you're testing is too high, that could affect the performance of the viability readout, which is why you should just use relatively fresh media. But the question is does that affect does the actual assay, the MTXD, affect the reading of the phenol red? I think with these assays, and Michael, you can elaborate on this. They're relatively so quick, and the whole point of these assays is it gives a color change, to show viability. So I don't think that's going to affect, over that short period of time. I don't think that's going to affect the acidity. I don't know. That would be my answer. I don't think so.
Yeah.
Okay.
Yeah, I'd have to agree with Steve on that.
Yeah, I would agree with as well. All right, here's an interesting question. Can reduction of CO2 for a limited time induce change in the cell's shape and behavior? And I don't know if they mean limited time, like you pull, your flask out of the incubator, put it in the biosafety cabinet, refeed the cells or do something like that, or leave them out and then put it back in. Or if you're talking about, leaving it out for an hour and putting it back in. Or the other scenario that I would think of is what happens if your CO2 tank runs out of CO2 and you don't hear the alarm or whatever.
Okay, so starting with the least amount of time, like taking out cells or taking out flasks to do media changes or passaging, things like that. I can't see that having any significant effect on the cells. It's common practice. You don't work with cells inside the incubator. You work with them inside the biosafety cabinet, prevent contamination and everything like that. So I can't see, that little momentary time out of the incubator could cause any problems. Getting up to an hour, I mean, if that's a little hard to say. I mean, are the cells being kept at room temperature for an hour? That's a little bit hard to say. And then when going into long term, like you suggested, the CO2 tank going out and not hearing the alarm go off, that can definitely have an effect on the cells. The biggest concern is going to be without that excess CO2, the media is going to become pretty basic. And extremely basic or extremely acidic media is not going to be great for cell growth. As far as things like change in cell shape and behavior, that's a little broad. That's hard to say. You can definitely might be seeing some cells detaching or apoptosing from the stresses caused from lack of CO2 for an extended period of time. But as far as changes in behavior, that's a little kind of hard for us to say if function-wise, if the cells are going to be different after not being in CO2 for that long. But cell shape can definitely be affected. Cell health can definitely be affected, sure.
Yeah, I guess the best remedy for that, or best precaution of that, is to, whenever your cells are out of the incubator, like for a feed or wash change, do it as quickly as you can. And if you're doing any experimental assay on, and make sure they're adjusted back to the incubator for some time before you do that. That would be probably, I guess, the best sort of response to that.
Okay. All right. Wow, we have a ton of questions come in, and we are over time, but I'd like to keep going if it's okay with Steve, Mike.
Yeah, I have no problem with that at all.
That can go on for some time longer, yeah.
All right, good. Thank you guys both for this. So, all right, is there any method of purification from bacterial contamination that you can do with a cryopreserved stock?
I mean, kind of like we touched upon at the beginning of the Q&A session, if something's contaminated, it's definitely better to get rid of it. One, because the effort required to purify a stock of contamination is really tedious. The cells may not even survive that. And then two, you've got the risk of contaminating other cells in your incubator. It's definitely just better to, if you know you have a contamination, get rid of the vial, get rid of the flask, and clean everything out.
Yeah, really. When in doubt, bleach it out.
Mm-hmm.
Okay. Well, how do you deal with cells that have very low viability and they don't seem like they're dividing or forming colonies?
So there are cells coming out. Excuse me. Cells coming out of cryopreservation tend to have can look pretty poor to begin with. Coming out of cryopreservation is very stressful on cells. Low viability and lack of growth, I mean, that could definitely be due to a lot of different factors. Again, one, stresses coming out of cryopreservation. Two, if the media is, what we recommend, if there are any differences there, it may take some time for the cells to acclimate to this new formulation. pH issues, contamination, mycoplasma, things like that. So it's kind of hard to say, kind of give a catch-all for everything that could happen because there are so many causes that could be happening there. But coming out of cryopreservation, sometimes with some cells, it is better to kind of just leave them alone for a few days, minimally handling them, trying not to do complete media changes, things like that. Basically, just giving them time to recover from the whole cryopreservation ordeal can help a little bit.
All right. Yeah, that makes sense. Give them a little rest after you wake them up.
Mm-hmm.
All right. This next question, basically, I'm going to try and wrap up several questions that have come in around this, but it has to do with generally the maximum amount of time that you can, or maximum number of passages of continuous cell lines and primary cells.
Okay. So primary cells, did you want to handle this one, Steve?
Yeah, I would say that for most continuous cell lines, there's no real limit. There could be some for specific ones that would be cell by cell, but for the most part, continuous cell lines literally are continuous. They can be passaged ongoing with no real limit. If you do have a continuous cell line, you're seeing issues where you passage it a lot and it's starting to conk out or so. You can the best thing to do is to go back to a fresh vial because there could be something in your cell culture procedure that could be causing that. So always be prepared to sort of start with the fresh vial. For primary cells, they almost all are limited to like 10 to 15 doublings, which is about 8 to 10 or more so passages. And once they get after about 10 to most of our primary cells, they'll go up to 15 doublings, but after that, they do start to lose their viability, their metabolism slows down. For primary cells, I wouldn't recommend sort of pushing it that far. I would keeping him less than 10 doublings and less than 6 to 8 passages or so forth. But Michael, please, you've covered probably these issues with lots more than me, so please add on to that if you think.
Oh no, that definitely covers a lot of it. So primary cells, like Steve said, finite lifespan, 10 to 15 doublings. You're going to really want to keep very sharp, close eyes on cell counts. Keep count of the doublings so that you can kind of know when to expect the cells to begin to senesce. Continuous cell lines, again, they should go indefinitely. In general, we do kind of recommend not going, I would say, more than 10 passages beyond, an original seed vial. Extended passaging can cause genetic drift. It can cause changes in, or rather, a selection of a subpopulation of cells. So, while they can go on indefinitely, for homogeneity purposes, it definitely is more worthwhile to kind of try and keep that within 10 to 15 passages from the original vial that's you created the seed stocks from.
Okay,
Yeah,
I like
I
it.
agree with that.
Now, somewhat related, since we're talking about passages, how often, for example, number of passages or period of time, do you recommend for mycoplasma testing and STR, if it's human or mouse?
So for STR, if for publication purposes, I assume you're going to want to send in, cells that are representative of what you used in the publication. So I assume probably the most recent passage. I'm not sure if there's a limit per se for that. I mean, as long as it's a human cell line, it should still be able to be picked up for STR purposes. Mycoplasma, I don't know if there's necessarily a passage recommendation for that. I mean, it can be tested for repeatedly, I assume, but I'm not sure if you would have anything to add on to that, Steve.
Yeah, I mean, I guess it depends on if you've passaged them, if you keep them in in your incubator while passaging them a few times, the more they're exposed there, the more likely they're going to come in contact with something. So if I would say, if you've got if you're going from say passage 2 to passage 8, I would say probably at least check it maybe one time in between. If you have some primary cells that are in your freezer, liquid nitrogen, you haven't worked with in a while, you don't really recall who tested it last. It's always good to when you first culture them to maybe take a sample then. So sort of initially when you start your cell culture, unless it's a commercially made a commercially available primary cell that you get from like ATCC, of course, where when you receive it, it's pretty much guaranteed to be myco-free. But if you've got cells, any kind of cells that you're ambiguous about when the last time it was checked, you should probably check it then.
Okay. Yeah, generally if your cells aren't performing correctly, you'll definitely want to do it then.
Correct. Yeah, if you see any kind of good point, if you see any kind of abnormality with growth, if they're not growing as fast or anything of that nature, definitely that should be something you should investigate with.
Treating, yeah,
exactly.
Not reacting appropriately to positive controls, etc. Okay. Now, let's get back to suspension culture. Is it necessary to keep the cells under agitation to keep them suspended?
No, especially for most smaller cell cultures, it's not really going to be needed. Agitation's really only going to be kind of a major concern when you start getting into bioreactor levels of volumes, we're going to be concerned about like air exchange for higher levels of volumes, but working in a T25, T75, there's really no need for agitation. You can do gentle rocking if desired, but there's really no requirement to keep the cells under constant agitation.
Okay. This next question is around trypsin. Why does trypsin sometimes not detach my cells? And maybe you could just generally discuss trypsin and what, having it on for too long or for too little time, what some of those effects might be as well.
So when you say that the trypsin's not causing the cells to detach, the first thing that immediately I would think about is excess serum in the culture. Washing cells with PBS prior to adding the trypsin can help with that. FBS is going to inactivate trypsin. That's why it's used as the part of the neutralization buffer. FBS traces in the culture could be affecting trypsin. The age of the trypsin could be affecting it too. If it is an enzyme, if it's kept at 4C for extended amount of time, you could see a lower amount of activity. If the pH of it is off, there are definitely factors there that could be affecting trypsin, but the first thing that I would definitely recommend is making sure that your cells are washed with PBS prior to the trypsin so that any trace FBS is not there that could interfere with it. And of course, some cells are just really strongly attached. Some cells are going to require 15 minutes plus of incubation in trypsin. Again, it's going to kind of vary cell line to cell line, but traces of FBS would be the first thing I would look into if you're having issues dissociating cells.
Yeah, I would agree with that. I'd only add on that specifically for primary cells, the trypsin recommended for primary cells is a more gentler trypsin. It has lower EDTA in it, and that will make detachment of primary cells even a little bit more difficult. So a general practice is be patient. Like Michael said, it's going to take, you may have to wait 15 minutes and do some general shaking and tapping on the bottom of the is needed for that. So if you were in the primary cells, you use a gentler trypsin, and you expect them to usually be a little bit harder to detach, or take a little bit longer to detach, I should say.
All right. Now I guess we'll do two more questions and close the Q&A session. So for this first question, is there any effect on preserved cells on genetic properties such as chromosomal aberrations? Can that happen when you cryopreserve your cells?
As far as I'm aware, no. I mean, the cells are going to have to be alive and proliferating for any sort of like recombination events to occur during mitosis and things like that. Chromosomal aberrations, like you will see in some spontaneously immortalized cell lines, that's going to happen over multiple generations, multiple passages. It's not going to happen as they're cryopreserved. They're going to have to be actively growing in culture before, you could even think about that happening.
Okay. Yeah, no, that makes sense.
Yep.
All right. And then for the last question, let's see. Let's find a good one here. All right. ATCC generally recommends growing cells without antibiotics, right? Do you have any recommendations of how to take care of the cells, how to culture them without introducing contamination?
Yep. The aseptic technique, aseptic technique, aseptic technique.
Yes.
Make sure.
Nice.
Make sure you've got gloves, lab coats, your inside of the biosafety hood is completely clean. You disinfect it after every use. Your aspiration needle is clean, that you don't not reusing any pipettes. Media is not being shared. I could go on and on and on, but aseptic technique, aseptic technique, aseptic technique.
And also make sure your incubator is cleaned out as well, and the same as the hood on a regular basis. Yeah.
Oh yes. Oh, and the hot water bath. The hot water bath.
And the hot water bath.
Yeah, that is the #1 place for contaminants to be introduced. Either use fresh water every time, or use a separate beaker with its own fresh water inside of it. Basically, try not to use dirty water. Dirty water leads to dirty cultures, and dirty cultures are bad.
Absolutely. Well said.
All right, I love it. I love it. All right, well, at this time, we will conclude our Q&A session. I'd like to thank Steve and Mike for the excellent presentation and fielding your questions. Also, thank you, everyone, for attending the webinar. Just a couple final words. We have some webinars coming up that might be of interest to you. On September 29th, Steve Budd comes back to discuss organoid growth media techniques to help you streamline your culture. It has a excellent demonstration video of how to make organoid growth media, narrated by James Clinton. We've got a couple of airway 3D airway webinars coming up soon. So on October 6th, Kevin Tayo is going to present evaluating the differentiation potential of primary airway cells in 3D models. He comes back again on November 3rd to talk about does differentiation matter, comparing the toxicological response between different airway epithelial models. And then on October 13th, John Folk presents his talk on luciferase reporter cancer cell lines, facilitate your CAR T development. And then finally, for a bit of shameless self-promotion, ATCC has a podcast called Behind the Biology. That's available now on Stitcher, Google, and all of your other podcast platforms, or ATCC website, www.atcc.org/behindthebiology. So thank you again, everyone, and have a great day.
Thank you.